Good morning everyone. In this video we are going to discuss about the neural tube formation. It is an important event in embryogenesis. The neural tube gives rise to brain and spinal cord. The whole of the nervous system is derived from surface ectoderm covering the embryo. While the embryo is still in the form of three-layered embryonic disc, the surface ectoderm and the dorsal aspect of the notochord differentiates. The part of the ectoderm destined to give origin to the brain and spinal cord can be distinguished as a specialized part called neuroectoderm. What is neurulation? It is the process of formation of neural tube by conversion of neural plate. It extends from pre-somite to post-time period of development. The neuroectoderm, it gives rise to neural tube, neural crest cells and ectodermal placodes. The neural tube gives rise to central nervous system and neural crest cell gives rise to most of the peripheral nervous system, and ectodermal placodes, it gives rise to cranial sensory ganglia, hypophysis cerebri and inner ear. This picture shows the dorsal view of pear-shaped embryonic disc with the slipper-shaped neural plate. The notochordal process, primitive knot, primitive streak and cloacal membrane. Next, coming to the stages of neurulation, neuroectoderm and neural plate stage and neural folding stage and stage of neural tube formation. First we see the neural plate formation. The surface ectoderm differentiates and thickens in the center. This thickened zone is called neural plate. The notochord acts as a primary inducer for neural plate formation and differentiation. The neural plate grows rapidly and elongates craniocaudally in the length. This stage occurs in 16th to 19th day. The next stage is neural folding stage. The continuous growth of neural plate makes it depressed in the midline. This linear depression is called neural groove. Elevated margins of the neural groove form neural folds. At the junctional zone of neural plate and surface ectoderm, the cells differentiate to form the neural crest. This stage occurs in 20th and 21st day of somite period. The next stage is neural tube formation. Due to rapid proliferation of neural plate, the neural folds come closer to each other and start fusion in the middle. On fusion, it forms the neural tube. The fusion begins in cervical region and extends in craniocaudal direction. The neural tube forms the central nervous system. This stage occurs in 22nd to 25th day. The stages in the formation of neural tube do not proceed simultaneously along the entire length of neural plate. The middle part is the first to become tubular around 21st and 22nd day, that is the 4th somite stage. The neural tube remains open at cranial end as cranial neuropore, that is called anterior neuropore, and at caudal end as posterior neuropore. The open neural tube facilitates circulation of amniotic fluid through the lumen of neural tube. It provides nutrition to a rapidly developing neuroectodermal cells before the establishment of uteroplacental circulation. The cranial neuropore closes by 25th day of intrauterine life, whereas caudal neuropore closes by 27th day of intrauterine life. Closure of cranial neuropore occurs at the 20th somite stage, whereas closure of posterior neuropore occurs at 25th somite stage. The non-closure of neuropore results in neural tube defects. The location of anterior neuropore is represented in adult by lamina terminalis, whereas posterior neuropore by terminal ventricle, that is lies in caudal end of spinal cord. Next, let us see about the differentiation of neural tube. The neural tube has a central cavity that forms ventricles of the brain and central canal of spinal cord, and a peripheral wall forms tissues of nervous system. The neural tube elongates craniocaudally. The cranial part of the cavity of neural tube dilates to form the brain vesicles, whereas the caudal part of central cavity remains tubular. The dilated cranial part of neural tube forms three primary brain vesicles that are called prosencephalon, mesencephalon and rhombencephalon. These are called forebrain, midbrain and hindbrain respectively. And further growth, 5 brain vesicles developed as follows. The prosencephalon divides into telencephalon and diencephalon. The telencephalon gives rise to two cerebral hemispheres The diencephalon gives rise to optic vesicle, pineal gland, thalamus and hypothalamus and posterior hypophysis. The mesencephalon gives rise to midbrain. The rhombencephalon divides into cranial metencephalon, caudal myelencephalon. The metencephalon gives rise to pons and cerebellum. The myelencephalon gives rise to medulla oblongata. Next, let us see about the flexures of the neural tube. The unequal growth rate of various components of neural tube result in flexures. There are four basic flexures of the neural tube. They are cervical flexure, cephalic flexure, pontine flexure, telencephalic flexure. The cervical flexure is situated at the junction of spinal cord and hindbrain. It makes 90-degree angle. The cephalic flexure, it occurs in the region of midbrain. It is concave ventrally. The pontine flexure at the junction of myelencephalon and metencephalon, it is convex ventrally. Next, the telencephalic flexure at the junction of telencephalon and diencephalon. Next, let us see about the cavity of neural tube. On the formation of brain vesicles, the cavity of neural tubes gets converted into ventricles and communications are as follows. The cavity of telencephalon develops lateral ventricles. The communication between cavities of telencephalon and diencephalon develops interventricular foramen of Monro. The cavity of diencephalon forms third ventricle. The cavity of mesencephalon forms cerebral aqueduct of Sylvius that connects the third and fourth ventricle. The cavity of rhombencephalon forms fourth ventricle. The cavity of spinal cord forms the central canal of spinal cord and its terminal ventricle. Due to rupture of roof, the fourth ventricle communicates with the subarachnoid space through foramen of Leschka and one central foramen, that is foramen of Magendie. That's all about the ventricles. Let us see a small recap of neural tube formation. Under the inductive influence of notochord, the neuroectoderm thickens to form the neural plate. The neural plate appears on 16th to 19th day. The neural plate folds along midline on day 20 to 21. At the end of third week, neural folds fuse to form the neural tube. Anterior and posterior neuropore closes on 25th and 27th day. Thank you.